Okay, we'll do attendance at the end. We'll do another one. Yeah. Good afternoon, gentlemen. So today we will start on the adaptive immunity, beginning with the antibody production by B lymphocytes. So the objective of this lecture is to talk about the basic general structure of antibodies, to recognize what is the part on the antibody molecule that's responsible for antigen binding, and that part which is responsible for giving it its biological characteristics, like killing antigen-bound targets, opsonization, what makes it IgG, IgM, etc., and so on. So these classes of antibodies, IgG, IgM, IgE, we call them isotypes. Then to talk about technological development that happened around the late 70s, namely the development of monoclonal antibodies, which is a big factor in immunotherapeutics these days. Then to describe how antibodies work in terms of removing the targets that they bind to, bacterial cells, viruses, etc. We probably won't have time today to talk about the nature of the antigens. We'll do that in the next lecture. So first of all, what are the antibodies? So as you should all know, they are made by B lymphocytes. There are two types of antibodies. One is an antibody receptor, which is essentially just an antibody molecule that is anchored into a B cell membrane. And that is the sensor or the activator which drives the B cell to make and then secrete that particular type of antibody, right? So basically you have a B cell with this antibody molecule linked onto it. As soon as it sees an antigen, you get cross-linking of those B cell receptors, and they stimulate that B cell to churn out that type of antibody, that particular antibody. So each clone of B cell will only make one particular antibody, one type of antibody. The antibodies recognize proteins. So in comparison, you get antigens of all different sizes, but generally the antibody is tiny compared to a bacterial cell. So an antibody molecule will only recognize what we call epitopes. Those epitopes are around 10 to 12 amino acids on that general antigen structure, whether it's a protein, whether it's a bacterial cell. When the B cell via the receptor has been stimulated on recognition of an antigen and makes antibodies, these B cells differentiate into what are called plasma cells. So these are fully dedicated cells that proliferate, and their only role is to make and release these antibodies. Once the antigenic load has been reduced, once the infection has been dealt with, most of those antibody-producing plasma cells will die, but some of those cells will remain alive and keep on growing. We don't know exactly where they reside in the body. We suspect somewhere in the bone marrow. And these cells can stay alive and keep on making antibodies for months or years after you've, say, had a virus infection. So they don't need antigen to keep stimulating those cells to grow. They can still, throughout your lifetime, you can still have some of these plasma cells around making antibodies to a virus that you might have been infected with when you were a kid. As I said, most of those B cells that were made to make antibodies during an infection die off, but the remaining portion will differentiate into these so-called memory cells. And these memory cells are the reason why vaccines are so effective, because vaccination generates memory cells. And these memory cells, as soon as you get exposure to, say, the virus that you are vaccinated against, these memory cells will immediately produce these good antibodies for blocking or neutralizing these viruses. And then finally, just a general, some general knowledge when you're working in a clinic, the study of antibodies and antibody levels is known as serology. So you'll often, in the clinic, when you take the patient samples, you'll often hear, let's take it to serology, let's take it to the serology lab to look at this person's antibody levels. So when you should all know this. This is all, again, general knowledge, but if you take blood from any organism and allow it to clot, this clear fluid at the top is serum, right? If you were to inject, say, a rabbit with a particular antigen like albumin, after 2 weeks, you take that blood and allow it to clot. This fluid, the serum, because it contains antibodies to that albumin, we call that antiserum. And in textbooks, you can see antibodies being referred to by different names. They might be called gamma globulin, they might be called immunoglobulin, they're abbreviated as IG, and we know them as antibodies. They're called gamma globulin because in the very early stages, before we even knew the existence of antibodies, what the early immunologists did was they took this antiserum and they blotted it on a membrane. And there's a lab technique called electrophoresis where you pass an electric current. So you have a negative, positive pole. So you pass an electric current across this membrane. And when you do that, you separate all the proteins out based on their size and the charge. The charge is based on the amino acid sequence of those proteins. If you do that, you can separate the serum proteins into 5 components. The major component is serum albumin. So most of the protein in your serum consists of albumin, just like you find in eggs, lysozyme, and so on. Then you get these other globulin proteins. And they found that this fraction, which migrated the fastest under this electric current, this is what gave protection against albumin. So this had some kind of protective proteins in it. So they call this immunoglobulin. And so this is where the antibodies, and this is gamma globulin. Now, the structure of all antibodies are the same. Essentially, they are mirror images of each other. They consist of 4 proteins. 2 which are much larger, which we call the heavy chain, associated with 2 smaller proteins called the light chain. And because this structure is symmetrical, it means that the amino acid sequence or the composition of one half of this protein is identical to the other half, right? And if you look at the amino terminal end of this protein, this is where the antigen binding occurs. So it means that this antibody molecule can bind to 2 antigens. It has 2 antigen binding sites. And because every antigen is different, those structures have to accommodate, or they have to be complementary to the antigen site. And so we call these the variable region, because that is where the variability of the DNA sequence of the gene that codes for these proteins is strongest. That is where you'll find the biggest difference in the genetic code responsible for these sequences, right? So essentially, all antibodies, 40% of those structures have similar genetic sequences. So the antibody molecule is essentially like a skeleton accommodating these variable regions responsible for binding to antigen, each antigen being different. The other region, yes?
The antibody, so for the variable side, that's where comes the antigen.
Yes.
And what makes it variable?
What makes it variable? That will come in the next lecture where I talk about immunogenetics. So what makes it variable? What makes it variable is, imagine a key fitting into a lock, right? So every lock has a different shape. That is the antigen. The shape is dependent on the amino acid sequence of that antigen, because the amino acid sequence will determine how that shape folds. So if you consider the surface of a virus with the proteins, you can have pits or like little hills on the surface. So in order for an antibody to recognize those shapes, it has to have the complementary sequence to be able to fit onto that. So it's all about shapes, because those shapes also, with those shapes come electrostatic charges between amino acids on the antigen and on the antibody binding site. So it's all about complementarity, right? Now, even though most of the skeletal framework is similar between all different antibodies, irrespective of their specificity, another change or another difference lies within this carboxy terminal end, which we call the FC region or the constant region. The sequence in this region determines whether that antibody is IgM, IgG, IgE, and so on. And it also confers to the antibody its biological properties, whether the antibody can fix complement, whether the antibody can bind to a macrophage in terms of opsonization, and so on. Also, I should mention, and this will become clearer when I talk about immunogenetics, is that there are two classes of light chain, one called a kappa and one called a lambda. So each antibody molecule will only be associated with one type of kappa chain or one type of lambda chain. This is what gives the antibody this variability. Yeah, so again, just to repeat, every B cell can only make one particular antibody of one specificity, right? Another thing I should mention, which you don't really need to know, but just out of interest, is that in the early days, when we first isolated these proteins with this antibody behavior, with the basic tools that the early immunologists had, they only knew about these particular enzymes which could cut proteins at particular amino acids. Using trypsin, you can cut the antibody here. When you do that, basically what that gives you is a shortened antibody molecule where you're removing this FC region, right? And so this shortened version still retains its ability to bind to two antigens, but because it's smaller, it means that this antibody can penetrate into cells, penetrate into tissues, and so this is another new type of therapeutic development with regarding antibodies. A very new development, a lot of research is going into the research on camelid antibodies, antibodies made by camels, llamas, all animals in that species, because those animals make much smaller antibodies compared to us humans and so on. And because they make smaller antibodies, there's a lot of research now looking at using these as therapeutic antibodies for COVID and so on. So that is the FAB region, and that is the FC region. There's another enzyme site over here where you can then cleave, so you'd get two individual FAB fragments. And so you'd only get one binding within these fragments. So that makes it even smaller. Now, what determines the specificity of the antibody? As I said, the antigen binding site lies in this region, the N-terminal end of the antibody molecule. And as I said, the genetic sequence of the genes that code for this protein determines the amino acid sequence, which determines how this structure shapes or how it fits to a particular antigen, right? I said that most of this is like a skeleton, so most of the genetic sequence will be similar between different antibodies. But if you look in this region and you look at the genetic coding for this region, this is where you will find the greatest variability between different antibody molecules. And because of that, we call this the hypervariable region. And these hypervariable regions, of which there are three on each chain, the light chain and the heavy chain. These three hypervariable regions are what determines the shape of this antigen binding structure. And so this is what gives the antibody molecule its specificity. These three regions are called CDR regions. So we have CDR 1, 2, and 3 on both the variable region of these light chains and on the heavy chains. So an antibody molecule then has got 12 CDR regions, 3 CDRs here, 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 and here. And these are responsible for recognizing and binding to the antigen structure. These CDRs look like fingers. So again, this is just one basic structure, but it might take other shapes. But this is what binds to the antigen, and this is what gives the antibody molecule its specificity, whether it's recognizing measles protein, bacterial surface protein, albumin, and so on. Now let's go on to this FC region, the constant region. So the constant region determines what type of antibody this is. We call these isotypes. So we have 5 isotypes: IgM, IgG, IgA, IgE, and IgD. Depending on that isotype and depending on that FC region will determine where those antibodies act in the body. So for instance, IgA is more closely associated with mucosal tissue, with the gut and the respiratory tract. IgA is actually the most abundant antibody that your body makes. But if you look in the blood, you'll find that it only makes a small component of antibodies. Most of the antibody in your blood is IgG. IgA is mainly in body secretions, in sweat, in tears, in the saliva, in mother's milk. So that gives an indication of the role of IgA is to trap antigens and then flush it out of the body, not to generate inflammatory response. I'll talk about that more next year when I talk about mucosal immunity. IgE, as a lot of you know, is associated with allergies, because when you breathe in pollen, IgE will bind to the pollen and then bind to mast cells and cause histamine release. Then two of these isotypes, namely IgM, well, IgG and IgA, have different subgroups there. So IgG has 4 subgroups in humans. Depending on which subgroup will determine whether the antibody can bind complement, whether it can generate strong inflammatory responses, and so on. Don't worry too much about those details. Going back to the antibody structure, you find that where the light chains link up with the heavy chain, you have cysteine residues here. And what do you know about cysteine? Creates disulfide bonds. So basically then, these cysteine bonds allow for the 2 jaws of the antibody to be flexible. And so this allows for the antibody molecule to engage via the variable regions, antigens which are either close together on a bacterial cell or whatever, or far apart. So this gives flexibility in that respect. Another aspect is that 2 of these isotypes, IgM and IgA, can associate with each other to form complexes. So with IgA, 2 IgA antibodies can join via the Fc region to form what are called dimers. And as a result, this dimer then can bind to 4 antigen binding sites. IgM can either form pentamers or hexamers. And in this case, then, in a pentamer, this IgM pentamer can bind to 10 antigens, and the hexamer can bind to 12 antigens. What's the significance of this? Well, first of all, with the dimer, it can cover a much broader area, much wider area. It allows for binding to more targets. And again, this will come in more detail when I talk about mucosal immunity, because these dimers get secreted into the gut, where they lie along the lumen of the gut and can trap antigens that you take into your gut and then get flushed out. The IgM pentamers and hexamers, you can think of them like innate antibodies in that they are first responders when you get an infection. And just like those nuclear extracellular traps I told you about with the neutrophils, these act like glues to trap any bacteria during an invasion. So now we need to understand 2 terms when we refer to the activity of an antibody or its sufficiency. And that is affinity and avidity. So affinity, think of a magnet or 2 magnets. You have a big magnet and a small magnet, right? So the bigger magnet has a stronger magnetic force. And that would be an antibody like IgG, highly specific for a particular antigen, because the complementarities between the antibody and the antigen has such a close fit that it binds with the strongest affinity. IgM has less affinity or specificity, but because IgM can form these complexes of 5 or 6 antibody molecules together, we say then that these pentamers and hexamers bind with greater avidity. So even though this pentamer has low affinity or doesn't have as great an affinity for antigen, it will bind with greater avidity because there are more bindings. So again, think of one big magnet that binds with a strong magnetic force compared to 5 or 6 little magnets that bind with less magnetic force. So with respect to the different properties of the different isotypes, just in general terms, IgM is the first antibody or the major antibody to be produced during a primary response. So the first exposure, the first infection you get with the virus or whatever, the first antibody you see is IgM. This is an important clinical biomarker. So this tells us whether the patient's been infected for the first time with a pathogen or has been reinfected. If we see IgG, we know it's a secondary response. So it means that the person got infected before, made IgM, but now it's getting exposed to the flu virus again, and it's IgG, we know it's a secondary infection. The characteristics of IgG is that, first of all, every protein that's made in the body has a certain half-life, right? And these are the basic half-lives of antibody molecules in the blood. So IgM, so you can see IgM, IgA, E, generally they're between 2 to 4 days. So once the antibody is made, it circulates and then it gets degraded within cells. IgG, on the other hand, can last for 3 to 4 weeks. How does it do this? It's because when these proteins, not just antibodies, but any protein in your body, when it goes past its use-by date, it gets taken into a cell, any cell, and gets degraded within lysosomes. What IgG can do is that there is a receptor on cells called the neonatal Fc receptor. And this is specific for the Fc region of IgG. This receptor will then take that IgG into the cell and bypass the lysosomes and push it out again. So basically, this Fc receptor, which is like a mobile receptor, is protecting that IgG from degradation within the lysosomes. So this is why it can persist and has a much longer half-life. The significance of this is regarding pharmacology, where they're now using this receptor to tag onto drugs to allow for longer half-life when they introduce these drugs into the body, so they don't get degraded in these lysosomes. The other significance of this Fc receptor, this neonatal Fc receptor, is that it allows IgG to cross the placenta to get into the fetus to provide immune protection to the fetus. IgA, as I said, is mostly in secretions and particularly in mother's milk. So the significance of this is that newly born babies are protected because when they get breastfeed, they're getting IgA from the mother, and that's giving it a certain level of protection.
Is there IgA on the skin?
Sorry?
Is there IgA on the skin?
It's yeah, but it's just shed, but it's in sweat.
That means IgG is only at about 4 weeks, 3 weeks, 4 weeks.
Yeah, not just in a mother, but all of us.
Does that mean the receptor that
No, that was where it was first discovered, but we now know that the neonatal FC receptor is in adults as well. That neonatal FC receptor was how the immunologists discovered that IgG could cross the center. But we now know that this is the general. As I said, IgE, you will learn more about IgE in when I talk about hypersensitivity, because it's associated with activating mast cells to release histamine. IgD, even though we've known about it since the 50s, we still don't really understand what IgD does, because it's mostly membrane bound in immature B cells associated with IgM, but it's so scarce in blood, we don't understand anything about it. So this is the types of antibodies you get if you were to be infected for the first time, or to inject an animal with an antigen like albumin. So you can see that after the first 7 days after the first injection, the first antibody you will see is IgM. And then a bit later, some IgG will come up. Then it will disappear. Now, if you give a second injection, you see that IgM levels stay more or less the same, but you get this huge upshoot in IgG. And that is because in this primary response here, you develop memory cells which were able to immediately activate and churn out huge amounts of IgG. And this is why vaccines are so effective in protecting us against virus diseases. So this is the difference between the immune response during a primary and a secondary infection, that the major antibody isotype made in a primary infection is IgM, whereas in the secondary infection is IgG. The detection of titers is relatively 7 days with regard to IgM, but in a secondary infection, 2 to 3 days, you'll see IgG. As I said, IgM is relatively low affinity, so it's like a trapping type of antibody, but the affinity of IgG is very, very high. And again, you get memory cells which can make IgG. And this is again why vaccines are so effective. So you get infected, say, with a virus, and even though you've been vaccinated, when you see the virus, when you get exposed to the virus properly, you make lots and lots of high affinity antibodies that can bind to the virus. So big deal. So now you've got this virus particle or this bacterial cell tagged with all these antibody molecules. So how does that bacterial cell or virus get removed from the body? By phagocytosis, by opsonization. Yeah, that's one mechanism. Complement, because once the antibody has bound to that cell, complement can bind to the Fc region of that antibody and then create this membrane attack complex that can kill or punch holes into the bacterial cell. What else? Sorry? NK cells, yes. Through this mechanism called antibody-dependent cellular cytotoxicity, which is similar to opsonization because both work through Fc receptors on the effector cell, recognizing the Fc region of the bound antibody. But unlike opsonization, which involves phagocytosis, the ADCC allows for the NK cell to get right up to the bacterial cell. And instead of phagocytosing, what the NK cell does is it injects these granules into the bacterial cell, which causes the cell to undergo apoptosis. Or this can also be a cancer cell, right? A fourth mechanism is in the case of viruses, which need receptors to attach and then infect host cells. So again, vaccine antibodies, which are specific for that receptor protein, can block that receptor from attaching to the host cell and infection. So this is what we call neutralization. This is just a description of ADCC. Finally, monoclonal versus polyclonal antibodies. So the conventional mechanism of making antibodies, like if you want to make antibodies against snakebite venom or tetanus toxoid or so on, is to inject an animal like a rabbit, sheep, monkey, donkey, whatever, with inactivated toxin. And after 2 weeks, you extract the antibodies from these animals. So you've got stocks of ready-made antibodies. That takes time. That takes 2 weeks for the antibody response to be generated and whatever. So a big technological breakthrough came in the late 70s with immunologists who were studying B-cell lymphomas. So this is a leukemic B-cell, which, because it is oncogenic, has cancer-causing genes, which allows these B-cells to grow forever. We call these immortalized cells, right? So what they did was they injected an animal to generate an antibody response, like you do to raise polyclonal antibodies. But then take out the spleens of these animals and isolate individual antibody-forming B cells. So they take out one antibody-forming B cell and they fuse it with one of these B-cell lymphoma cells. And that fusion product imparts these cancer-causing genes to that antibody-forming B cell to make that cell immortalized. And so in this way, you can grow liters and liters of these B cells, making this one particular antibody. And this is what you can buy from drug companies. This is the major monoclonal antibodies that you use for immunotherapeutics. which you'll learn later in pharmacology. So the difference between a polyclonal and a monoclonal antibody is because the polyclonal antibody has been generated from a whole animal, from multiple B cells. You have a wide selection of B cells making antibodies to the same antigen, but different specificities directed against different epitopes. With a monoclonal antibody, you only have one particular species of antibody recognizing one particular epitope. And the applications of these monoclonal antibodies, as I said, we use them for immunotherapeutics to treat cancer, arthritis, autoimmune diseases, etc. It's a big value in terms of research, because by tagging these antibodies with chemical dyes, we can identify cells or tissues or antigens in tissues using a UV fluorescence microscope. We can also tag the FC region with magnetic beads. And so if you incubate a cell mixture where you want to isolate a particular protein or whatever, you then just put a magnet to this side of the test tube and it'll pull out all of those bound antibodies. Okay, so we'll leave it for now, and then I'll talk about antigens in the next lecture. You want to take attendance again, or is everyone done? Yeah, I got the.
So I took attendance in the beginning, and I took it again just to make sure, and it made me take it again. So that means this time counts. I guess.
What's your name?
Badr Gahtan. Yeah, I was able to take it twice. So that means the old one wasn't safe, maybe.
Fadl.
Badr, BA.
BA.
Yes, that's it.
Okay.
Oh, yeah. Thank you.
Yeah. Hi, you want to stop?
I love you.
Okay.
Thank you, doctor.
Okay. Thanks.
Have a nice day. And let's do our thing. End meeting.